G'day guys, and welcome back to the third part of this presentation. It's Dr. Buddhamananiakara, clinical lecturer at Charles Sturt University and respiratory physician at Orange Health Service. So, we'll move on to the third part of our presentation. So, we've now learnt that oxygen is absolutely vital for aerobic respiration, so much so to generate the significant quantities of ATP. We've also learned a bit about the oxygen cascade, how oxygen comes in from the ambient environment, and we calculated the partial pressure of oxygen using some of the gas laws that we learned in dealing with partial pressure. We described how gas exchange occurs in the alveoli and that oxygen diffuses down its partial pressure gradient into the pulmonary circulation, from which it's transported by mass convection or blood bulk flow from the heart into the tissues, where the ATP is eventually utilized in the mitochondria. The oxygen is utilized by the mitochondria to produce the sufficient quantities of ATP. So our next part will talk a bit about oxygen solubility, and it will generate a problem for us to think about. Essentially, what I want you guys to understand here is that oxygen is extremely poorly soluble in blood, and to solve this problem, in order to deliver enough oxygen to make the ATP through aerobic respiration, we need a carrier protein, and that carrier protein is called haemoglobin. So let's have a look at this in more detail. So we're going to introduce another gas law, and this gas law is Henry's law. And I want to have a look at this beaker here. So this is a beaker with a blue liquid, and above that blue liquid is a red gas. Now, Henry's law states that the concentration of gas, so these red particles, in solution, so interacting with the liquid molecules of the blue liquid, is directly proportional to the partial pressure of this gas here above the liquid times the solubility of the gas. And that kind of makes sense. If you think about it, if you have more gas here, so more, if you imagine these were red particles here, sorry about drawing in blue pen, then you could imagine that randomly more of these gas molecules will interact with the liquid and subsequently go into solution. So I think it should make sense that if the partial pressure of the gas increases, so too does the concentration of gas in solution. Also, if the intrinsic solubility or the tendency of the gas to associate with liquid is higher, then there will be more gas molecules in liquid phase. Let's have a look at this. So here we go. What we've done is we've doubled the partial pressure of gas simply by doubling the number of gas molecules. And what we see is that there's now double the amount of gas molecules within the same volume in concentration. Therefore, by doubling this, we've also doubled the concentration of gas in solution. Now, let's have a look at the problem. Well, you should all be familiar with the graph on the right here. Now, this is the graph of the oxygen cascade. We understood how we derived 160 millimeters of mercury of oxygen. That's simply by knowing that the barometric pressure is at 760 millimeters of mercury, and that oxygen occupies 21% of that. We then learnt that when you breathe it in, the air becomes humidified, and necessarily that dilutes the oxygen there, so that decreases it to 150 millimeters of mercury. And then we understood that when we get to the alveolar space, there is carbon dioxide and oxygen gas exchange. So that further dilutes the oxygen within the alveoli. We note that in the pulmonary capillaries, where oxygen goes in from the alveoli down a very thin membrane for diffusion, there is no change in oxygenation simply because the diffusion barrier is so thin, so there's no diffusion block. And then we learnt that when we get to the arterial blood here, that due to some venous admixture, remember through the bronchial arteries, thespian veins, PFO and other pulmonary arterial venous malformations, that the shunt is about 5%, so that there is some venous admixture with deoxygenated blood. And then we also realise that oxygen extraction occurs within the tissue and drops the oxygen content in venous blood, which is the effluent, from the tissues to 40 millimeters of mercury. Now, if we have a look at what's happening in the pulmonary capillaries, remember oxygen's diffusing down its partial pressure gradient from the alveoli into the pulmonary capillary. To have a look at the concentration of oxygen in solution, because after all, we need the oxygen to be within the blood in order for it to be pumped by bulk flow from the heart through the circulatory system into the tissues. It needs to be within the blood. So this is absolutely important. Now, we know that oxygen solution by Henry's law is proportional to the solubility and the partial pressure of oxygen. Now, we know that the partial pressure of oxygen is 100 millimeters of mercury. We've derived that as previous. But the solubility of oxygen in blood is very minimal. It's 0.003 mils per 100 mils per millimeter mercury of partial pressure. And that means that for 100 mils of blood, we only have 0.3 mils of oxygen. Now, the heart is able to pump 5 liters of blood per minute. That's the cardiac output, how much blood is pumped out per minute. And so we can find out how much oxygen is delivered based on dissolved oxygen. So we know that the oxygen in solution is 0.3 mils per 100 mil. That was derived from here. Now, if we have 5 liters per minute, converting it to mils per minute, cancelling these units out, we get that 15 mils of oxygen is delivered per minute. That's quite small, considering that for our resting basal metabolic needs, we need 250 mils of oxygen per minute to sustain it. And we know that why we need oxygen, simply for the purpose of aerobic respiration. If we don't have oxygen, we don't have ATP. We must rely on anaerobic respiration, which does not generate sufficient quantities of ATP. Relying on dissolved oxygen itself, we only get 15 mils of oxygen. We need 250 mils of oxygen. This is going to be a significant oxygen debt. So how do we solve this? And it transpires that we have this beautiful protein, probably arguably the most elegant protein that's developed in evolution, is hemoglobin. And the purpose of hemoglobin is to carry oxygen. And we'll go through how it does that later. But suffice to say that hemoglobin is so vitally important to animals that really there is only one animal in the world that does not have hemoglobin, and that's the Antarctic ice fish, and we'll explore that a little bit later because its story is quite interesting. Now we'll go over the molecular detail of hemoglobin in the next structure, but suffice to say that oxygen is carried in this group here, which is called the protoporphyrin ring, and it's complexed to iron, so it kind of forms a complex with iron there. And this protoporphyrin ring is bound within a protein subunit, and there are 4 protein subunits here, 2 alpha and 2 beta. Now, how does this solve it? Well, scientists have calculated that 1 gram of hemoglobin, when it's fully saturated with oxygen, when it's carrying as much oxygen as it can carry 1.3 mils of oxygen. What do we know? Well, we also know that the normal hemoglobin concentration in blood is 140 grams per litre. So, if we assume that oxygen is fully saturated, then we can work out at maximum how much oxygen a litre of blood can carry. So we know that we've got 140 grams per litre of haemoglobin, and we know that each gram of haemoglobin can carry this much oxygen, 1.39 mils per gram. So we know that each litre of blood can carry 194.6 mils of oxygen. And if we assume that we have 5 litres per minute of cardiac output as per previous, then it should follow that the delivery of oxygen with the addition of haemoglobin can produce 973 mils of oxygen per minute. That is vastly more than 250 mils of oxygen per minute required for basal metabolic rate, and much more greater than the 15 mils of oxygen per minute derived from dissolved oxygen. So I hope that makes sense, that oxygen is poorly soluble in blood, and we've talked about Henry's law, and it must be carried by haemoglobin to support the requirements of oxygen. Haemoglobin is a crucial protein.